I'm going to present the paper name Interpreting in vivo calcium signals from neuronal cell bodies, axons, and dendrites. This is the review paper, and this paper is about how to interpret in vivo calcium imaging signals in relation to the underlying electrophysiological events. So the calcium imaging has rapidly become a widely used technique in neuroscience over the past few years. The main reason for its popularity is that calcium influx within neurons reflects underlying electrical activity. And this,
Soon. Could you use a laser pointer when you're presenting? Okay,
okay.
thank you.
And the purpose of this review is to interpret what the fluorescence signals observed in different compartments, such as soma, axon, and dendrites, represent and which electrical events they correspond to. So the figure 1 describes the experiment that quantitatively examines how accurately the in vivo somatic calcium signals reflect neuronal spiking activity. So here, panel A, GCamp6F and GCamp6S was expressed, and two-photon microscopy was used for imaging while spikes were simultaneously recorded using juxtacellular electrodes. So here the above one, this shows the calcium signal, and this below one shows the electrical signal, the action potential. So fluorescence signal increased whenever spikes occurred, like here, indicating that the amplitude and pattern of the calcium signals correspond to the firing rate. And panel B, it shows the magnified view of the region, indicated by the dashed line here and here. And the clear observation that the fluorescence sharply increases each time a single spike occurs. And GCamp6S shows the slow rise and decay, whereas GCamp6F exhibit faster kinetics. And this illustrating the kinetic differences between the two indicators. And panel C shows the average fluorescence response obtained by aligning and averaging multiple single spike events. And GCamp6S exhibit a slow and prolonged waveform, whereas the GCamp6F shows a fast waveform. And this indicates that GCamp6 has a higher temporal resolution, making it more suitable for detecting the single spikes. So this figure is trying to examine how reliably somatic calcium signals reflect neuronal spiking activity. And by this experimental evidence, we can conclude that somatic calcium signal provide a reliable optical proxy for neuronal spiking with fluorescence of amplitude and time course tightly coupled to action potential generation. 혹시 빠른가요?
Yeah, every slide, why don't you ask question and people write questions down the chatting and you can wait for a minute, okay?
일단 넘어가겠습니다. So figure 2 shows the experiment designed to verify whether somatic calcium signals can reliably reflect spiking activity even in neurons with high firing rate. The panel A shows the simultaneous measurement of calcium signals and electrical signals in PB interneurons. So here the target is the parvalbumin expressing interneurons in layer 2/3 of the mouse visual cortex, and cells were identified by TD tomato expression and loaded with the synthetic calcium dye Oregon green bata 1. So here you can see the PB interneuron by stained with expressed with the TD tomato, and cell attached patch recording captured spiking activity while 2-photon imaging measured a fluorescence. So here, like dense part, fluorescence rises whenever spikes occur, and the magnitude increases with spike count. So it means somatic calcium signals closely follow the firing rate. And panel B is illustrate the relationship between the number of the spikes and the fluorescence amplitude. As the number of action potential increase here, if the number of the action potential increase, then the fluorescence signal increases too. And it demonstrates the linear relationship between calcium signal amplitude and spike count. And panel C shows the differences in calcium response among different neuron types. And PB and the somatostain interneurons show smaller calcium responses than the pyramidal neuron, the black one. And it's because of the calcium buffering. However, all the cell types display a rapid fluorescence rise following each spike, and it indicates that the somatic calcium signal represents a spiking activity. Panel D, as you guys can see, for all neuron types, higher firing rate produce proportionally larger calcium signals. So this section investigates whether somatic.
Sungjun, there's a question for that.
Mm. Mm. The when we use the Oregon BAPTA and they overlay the Oregon BAPTA as I know, the wavelength is different because the Oregon BAPTA for the filter for the Oregon BAPTA and the TD tomato, we need the different filter for visualize the cell. So that's the reason why the PB Oregon BAPTA image is more darker.
Sungjun, I think you may misunderstand his question. So that panel A OGB1 versus parvalbumin OGB1, the signals are different, but you already explained in panel C, second bullet, that the calcium responses are different from different types of neurons. Okay. So I guess that's what probably Sion was asking. But let me ask here maybe to comment. So let's look at that, this panel B. I think this is important. This shows in ideal case, number of action potentials will accumulate. So dF over F will be like kind of proportional. That would be ideal. That means just by observing the image, we could count action potential. But the green here is mean, so that is fine. However, the raw data of each of these actually show how much variable it is. So that means it probably is not possible from just dF over F to estimate the action potential, just because this relative signal strength are quite diverse between different cell type or even different positions. So I guess this is kind of a key reality I think we should be aware of. Okay. I'm not sure this is my interpretation from this very good raw data slide. Okay. Okay. I think we can move on. Yeah, thank you, Sunju.
네. And here this section investigate whether somatic calcium signals can reliably represent neuronal activity in cells with high firing rates, such as fast spiking GABAergic interneurons. And calcium transients are relatively slow compared to millisecond scale spikes. It questions whether optical signals can still track rapid firing dynamics without losing temporal accuracy. By this experimental evidence, we can conclude that somatic calcium signal can reliably represent overall firing activity in fast spiking neurons, even though individual action potential cannot be temporally reserved. And figure 3 presents the experiment designed to verify whether calcium imaging can sensitively reflect not only increases but also decreases in neuronal activity. So pattern A shows the reduction in calcium signals following neuronal inhibition. Here, the target cell is the layer 2 over 3 PB interneurons of the mouse visual cortex. And each cell expresses two key constructs, JR camp P1A. This used for the calcium imaging, and GTACR2 is used for the optogenetic inhibition. So fluorescence and juxtacellular spike activity were recorded simultaneously. And here, when we illuminate the light, which wavelength is 920 nanometer, the GTACR2 activation suppressed the neuronal spiking. As the firing rate decreased, the fluorescence signal also dropped, demonstrating that somatic calcium signal can report decreases in neuronal activity, not only increases. And panel B illustrate the calcium responses to change in firing rate using a Poisson spike tree simulation. Here, this part is showing the firing rate, and here left part, the frequency changed from 1 to 0.5 and come back to the 1 hertz. And middle one and the right one is same as here, but the number is different. Each spike generated the calcium transient model as a single exponential decay. So both here histogram and simulated calcium signal traces showed clear decreases during the reduction in firing rate. And averaging across 20 trials reproduced the same trend, confirming that the model captures the integrative and slow decay nature of somatic calcium signals.
Sungjun,
So,
can you explain what is Poisson spike train?
ah,
Yeah.
so this Poisson spike train simulation is, uh, we often use this simulation to make the random data. So, but the average should be
Mhm.
uh remain. 아니, the average should be the maintained. So we usually use this for the random data that the average is maintained average. Is it? Uh, mm.
Okay, then why did they use Poisson spike train here for simulation?
Mm, they, as I remember, they didn't mention about why they use this simulation tool, but I think they should uh, mimic the nature situation, and the firing rate should be the random firing should be occurred by randomly. So I think they use the Poisson simulation.
Yeah, that's a good answer. Yeah. So the Poisson distribution or this Poisson spike train is a random event mimicking the interspike interval is random, but the overall mean value remains the same. As you explained, the mean value should be the same as a square of standard deviation, okay, or variance. So that's mathematically how they generate. Okay, yeah, I think.
So this section explores whether somatic calcium signals can also represent decreases in neuronal firing rate, not just increases. And by this experimental evidence, we can conclude like somatic calcium signals can faithfully reflect decreases in neuronal firing rate, making them reliable bidirectional indicators of neural activity. Oh, and figure 4, the present experiment designed to examine how quantitatively external calcium signals reflect presynaptic activity, specifically spiking at the synaptic terminals. And panel A shows the experimental setup. The target cell is the long-range neurons connecting the retrosplenial cortex and CG1 and 2. And virus were injected into the RSC to express the GCAMP6S in RSC neurons. And two-photon imaging was performed in the CG1 and 2 cortical area, which received long-range external projection from RSC neurons. The calcium signals measured represent presynaptic activity originating from RSC neurons, terminating in the CG1 and 2 region. And panel B shows the in vivo imaging result from external photons. And the top panel shows the two-photon image of the GCAM6S expressing external segment in CG1 and 2. And the bottom panel displays the fluorescence trace from the button in response to either 1 or 16 electrical stimulation pulses. The single pulse produces a small fluorescence trisent, whereas 16 pulses cause the much larger fluorescence release. And this indicates that external calcium signals scale with stimulation strength and firing frequency. And each trace represents the mean of the 5 repeat trials. And panel C shows the changes in fluorescence intensity according to the number of stimulation pulses. As you guys can see, the number of pulses increase, then the fluorescence also increases. Fluorescence amplitude increases nearly linearly with personal number, indicating that external calcium signals quantitatively encode the level of presynaptic activation.
Oh, Sungjun, there was a question. Mhm. Mhm. Oh, every student should show their face, okay? Yeah,
Uh, as I understand,
yeah.
you are asking they perform the experiment, Poisson simulation, and electrical uh, signals together? 질문을 이해부터 했습니다.
효정이가 다시 질문할래?
So, um,
Yeah.
I was asking how the Poisson spike train simulation is done. So, my intuition was that they convolved the Poisson distribution with electrical signals so that they can express a kind of delay that was caused by the repetition of spikings. And yeah, I was just wanted to confirm my idea on that.
I think the answer is yes, but uh, I will search more and will answer. Can I answer later?
Yeah, thank you.
Yeah, it seems like the time seems to be over, so let's keep moving on. But write down all the questions. If there's a no answered one, then he can actually do it later in our Slack channel.
So this section examines whether external calcium signal can serve as reliable indicators of presynaptic neuronal activity, and by the experimental evidence, we can conclude like external calcium trisent faithfully represent presynaptic activation strengths, enabling quantitative optical measurement of long-range synaptic output. And here, figure 5 presents an experiment designed to examine how accurately external calcium signals in dopamine neurons reflect actual neuromodulator release, specifically dopamine release. So panel A shows the experimental setup. The target is the dopaminergic neurons located in the ventral tegmental area and substantia nigra. And pre-dependent G-CAMP6F was injected into the dopaminergic neurons. And the medial forebrain bundle, MFB, was electrically stimulated using the bipolar electrode to invoke dopamine release. And optical fiber and the carbon fiber microelectrode were implanted into nucleus accumbens and the dorsal-medial striatum, respectively, for simultaneous fiber photometry and cyclic voltammetry, for the extracellular dopamine concentration. And these two reasons represent distinct projection targets of VTA, SN dopaminergic neurons, allowing comparison of external calcium and dopamine dynamics between long-range output pathway. And panel B shows the vivo imaging result from, figure B displays the measurement of dopamine release using cyclic voltammetry. And here, the rise in oxidation current following stimulation confirmed dopamine release at the recording site. And panel C shows both calcium and dopamine signals. And both signals rise sharply following stimulation and reach their peaks nearly simultaneously. This temporal correspondence indicates that dopamine release is directly coupled to presynaptic calcium influx in dopaminergic axons. And panel D presents the latency to peak for each signal. And with the VTA SN to NA projection shows the shorter latency to peak compared to the VTA SN to SDMS projection. And panel E shows the duration during which signal remains above half of its maximum value. And the half-width of both fluorescence and dopamine present is significantly shorter in the NAC pathway than in the DMS pathway. And this suggests that NAC terminal have faster rise decay kinetic, whereas DMS terminal shows the slower and more prolonged signaling, possibly due to sustained release or slower reuptake. Panel F illustrates the relationship between stimulation intensity and calcium signal amplitude. When the increasing the number of stimulation pulses caused the gradual increases in fluorescence amplitude, the magnitude of external calcium responses correlated linearly with the measured dopamine concentration across both NAC and DMS projections. And this demonstrates that external calcium signals quantitatively reflect the amount of neurotransmitter release. And this section investigates whether external calcium signal can directly represent the release dynamic of neuromodulators, specifically dopamine, rather than only reflecting electrical spiking or presynaptic activation. And by the experimental evidence, we can conclude the external calcium detergent acts as reliable correlates of neuromodulator release, linking presynaptic calcium dynamics to the strength and timing of dopamine output in vivo. And figure 6 presents the experiment designed to determine how well dendritic calcium signals reflect somatic backpropagating action potentials. And panel A shows the result of the stimulating the soma while simultaneously measuring calcium signals and electrophysiological activity in the dendrites. And target cell was the layer 2/3 neuron in the visual cortex. And the cell was loaded with the calcium dioregon green beta and simultaneously recorded using a whole cell patch clamp technique. And here the green dashed line indicates the analyzed dendritic regions. And here the bottom trace represent the current injection applied to the soma, and red trace shows the corresponding membrane voltage. And here the fluorescence trace shows calcium transient recorded from the soma and dendrites. The amplitude of dendritic calcium signals increased linearly with the number of evoked spike, and dendritic calcium responses faithfully reflect the number of backpropagating action potentials. And panel B illustrates the changes in calcium signals according to the number of spikes. The x-axis is the number of the action potential, and y-axis is the mean dendritic calcium amplitude. And the fluorescence amplitude increased nearly linearly with a spike number, quantitatively confirming that greater backpropagating action potential activity produce a larger calcium influx in the dendrite. And panel C shows the differences in calcium signals depending on the distance for the soma. The top image shows the dendritic branch segmented into the recording sites, and bottom traces shows the plot the fluorescence signal from each site. And calcium transient were strongest near the soma and gradually weakened at more distal location, and indicating that the amplitude of the backpropagating action potential attenuates with distance along the dendrite. So this section explores how dendritic calcium signals can serve as an optical indicator of backpropagating action potentials. The electrical signals that travel backward from the soma into the dendritic tree. And by this experimental evidence, we can conclude backpropagating action potential evoke dendritic calcium signals that scale with spike number and decay with distance, offering a direct optical signature of soma to dendrite electric coupling. So before presenting the introducing about the figure 7, let me briefly explain the two types of the dendritic events. The first spontaneous dendritic voltage event is a small, naturally occurring sub-threshold voltage fluctuation in the dendrite. It's usually caused by the random or asynchronous cinetic inputs and does not trigger an action potential or a large calcium influx on its own. And in contrast, the regenerative dendritic event, also called the plateau, uh, plateau is the nonlinear and self-sustaining depolarization. And it happens when multiple synaptic inputs arrive synchronously and in a specially clustered way, activating voltage-gated calcium channels. And this leads to the long-lasting plateau depolarization and the large calcium signal. And to reproduce this experimentally, the researcher injected the weak sustained depolarizing current into the soma, and this by itself did not evoke calcium signals, but it biased the membrane potential. When the spontaneous dendritic events occurred at the same time, the two input cooperatively reached the threshold, triggering a regenerative plateau potential.
Got a question. Can you go back one slide? Quick question. So what does this mean? Do you understand? Could you explain? Backprogramming action potential evoke dendritic calcium signals that scale with spike number and decay with distance. So what does it do? Well, what's the mechanism?
So if we simulate the soma, and if the calcium signal in the dendrites increased, then we can define that it's because of the backpropagation.
Okay, but it just increases like the number of spikes, just so going back and forth, like it seems like, right? So more firing, more backpropagation, more firing, more backpropagation. Is it right? Okay.
Yes, because the
Okay.
here,
Then next slide. Then I'm just saying trying to understand what's going on. So next slide, and you said uh next slide. Yeah, and then you said there's the self-sustaining depolarization. Okay, so my question would be, so if that, okay, like it's like it feeding each other, more backpropagation, more spiking, more backpropagation, I guess. Is that
yes,
correct?
yes,
Then would it reach the self-sustaining depolarization anyways?
어, 질문을 이해를 못했습니다.
그러니까 backpropagation이 늘어나면은 스파이크 막 늘어난다고 했잖아, 그지?
네,
스파이크 늘어나면 100% 늘어나? 그렇겠지?
네,
그러면은 그게 계속 왔다 갔다 할 거 아니야, right?
네,
그래서 non-linear self-sustaining depolarization occurs when multiple synaptic inputs arrive synchronously. 그러니까 backpropagation이나 이게 multiple 시그널이 인풋이 되냐 이거지. Do you understand my question? 소마에서
네,
backpropagation이랑 계속 오는 인풋이랑 계속 만날 거 아니야, 그러면. 그지?
네,
Would it reach the self-sustaining depolarization by increasing backpropagation. Is it possible? Is it a possibility? 그게 가능해?
그러니까 제 교수님의 질문이
야, 교수님? 이 새끼야,
네.
넌 임종묘냐, 이 새끼야? 임종묘를 이름 붙이고.
네, 단장님의 질문이 소마를 자극을 하면 백프로파게이션이 계속 일어나고 백프로파게이션에 의해서 이게 계속 일어날 수 있지 않냐라는 질문인가요?
그렇지. 근데 이제 사실은 그건 좀 바보 같은 질문, 어떻게 보면 바보 같은 질문인데 이게 그래서 actually I'm trying to yeah, say that like when it occurs, does it really is it okay natural condition?
네.
이게 natural condition이 아니면 이 사람들이 실험할 때 이렇게 만들어낸 거야. self-sustaining
어,
플랫폼 같은 것들이.
실험할 때 만들어냈고
음.
백프로파게이션 같은 경우는 이 논문에서 사실 언급을 했었는데 백프로파게이션이 일어나지 않게끔 그 자극의 강도를 굉장히 낮췄습니다. 그래서 자극 강도를 굉장히 낮춰서 소마를 그 정극을 이용을 해서 소마를 자극함과 동시에
Okay,
그
so
네.
I understand that. So, but compared to real life,
네.
how much is different? I mean, I think we have to know what's the difference between the real life and then this scenario.
음,
내 생각, okay, so I joined in the middle of a thing, so I never read this article, but it sounds like uh, this is only possible in this under this condition. artificial
어,
같은 느낌이 들거든.
근데
응.
저는 이제 충분히 자연적인 상황에서도 일어날 수 있다고 생각을 하긴 하거든요. 왜냐면
응.
그 애초에 그 자연적인 현상을 미믹을 하려고 했는데 그런 거를 미믹을 제대로 할 수가 없는 환경이기 때문에 소마에다가 인위적으로 자극을 했다라는 뉘앙스로 이해를 했거든요.
그러니까 나는 I mean, I just don't understand if it is a real life event. Okay, no, but this is my question. So I'm kind of clear. For me, 나는 개인적으로는 I'm clarified. I don't think they had the in vivo
음,
data for this, right?
전부 다 인비보 데이터입니다.
100% 아까 자극 자체가 다, 그러니까 sorry, natural condition 아니라, 그러니까
네,
그 자극 자체가 다 artificial하게 준 거잖아.
네, 네,
그 말이야. Okay, sorry, okay,
네.
okay.
네, 넘어가서. So Figure 7 presents the experiment designed to investigate how dendritic calcium signals can be driven not by simple backpropagating action potentials, but uh, by intrinsic dendritic generative events, specifically plateau potentials. So panel A shows the experimental setup, and the target was the layer 5 pyramidal neurons of barrel cortex. And in vivo to photon imaging and dendritic patch clamp recording experiment performed. And membrane potential was recorded directly from the apical tough dendrite. And the calcium fluorescence was simultaneously measured in the same region. And panel B displays the actual imaging view, and the red line here indicates the location where calcium fluorescence was measured. And this region corresponds to the distal apical tuft dendrite or from the soma. And panel C here shows the voltage and calcium traces. The very top one, top trace shows the recording without any current injection. And we can see some spontaneous dendritic voltage image events, but they don't lead to the calcium signals. And the bottom three traces shows the result when the small depolarizing current was injected into the soma. Here, by itself, this current wasn't strong enough to trigger a back-propagating action potential or calcium influx. However, when the spontaneous dendritic event happened to occur at the same time as the somatic depolarization, they work together to produce the regenerative plateau potential and the large sustained calcium signals. And panel D presents the magnified view of the voltage trace at the dashed line box. And trace 1 and 2 shows the spontaneous dendritic voltage event recorded without current injection and producing small calcium signals. And trace 3 is a spontaneous dendritic event coinciding with somatic current injection, producing a plateau potential and strong G-camp response. And the gray trace indicates the spontaneous dendritic voltage image events, while the black traces represent coincident events occurring during somatic depolarization. And this section examines whether dendritic calcium signals can reliably represent locally regenerative electrical events that occur within dendrites. And by this experiment evidence, we can conclude dendritic calcium transient can cause non-linear regenerative dendritic events that occur independently of somatic firing, but distinguishingly truly local from globally propagated activity remains experimentally challenging. And last, this is the conclusion. In vivo calcium signals reflect diverse underlying electrical process that depends on neuronal compartment and context. Somatic calcium transient reliably report firing rate and excitability, though temporal precision is limited. And external calcium signal correspond to the presynaptic activity and neuromodulator release, representing neural output dynamics. Dendritic calcium signals arise from both back-propagating action potentials and locally regenerative events, indicating nonlinear synaptic integration and plasticity rather than spiking. And interpretations requires recognizing that calcium transients can encode output, input integration, or local computation, not a single uniform measure of activity. Therefore, calcium imaging provides the compartment-specific window into neural computation, linking electrical signals to behavior and network function. 네.